Okay, are we ready? Yeah. Okay. So there's no need for clapping right now. I'm sorry. Sorry about the delay. We had a bit of a mishap with the transportation, so that's why there was a delay, but we still have enough time. So this is Zachary Lewis. He's a PhD candidate at Harvard University. And he's actually going to tell us about evolution. We all know that we have learned about evolution. It's a very important part of our curriculum. So I know for my students, I will use this as a lesson and then we could get tested on it. This is a very important concept. So please make sure that you pay lots and lots of attention. Very lucky that you're actually getting a lecture on something that you have to learn from our Harvard graduate students. Not very many people can say that. So I want you to give him as much attention as you can. Ask him lots of good questions like you always do. So without further ado, we'll just hand it over to Mr. Lewis.
Thank you for having me and having me here. Can you guys hear me okay? Yeah. Okay, great. So up at the front, I don't think she knew she was going to be under this much pressure, but on the video camera is a salamander, and I'll talk about her later. And I've got a couple other animals up here as well. But yeah, if she starts freaking out or anything during this talk, let me know. I think she should be pretty quiet and still. So that's an axolotl, and we'll talk about her a little bit later. But I'm talking today about evolution and development. I didn't know you guys were going to be quizzed on this or tested, and I'm sorry for that. But I'll do my best to try to explain it. So Evo is this term that we use to talk about the combination of two fields, which is evolution and development. And you've all learned a little bit about each of these fields. And so I'm going to give you a bit of an introduction first, and then talk about the combination, the science that I'm interested in. First, I wanted to talk to you about myself a little bit and introduce myself. I'm originally from Seattle, Washington, or outside of Seattle. I went to college in Portland, which is not far away. At Reed College, and I studied there. I took some detours along the way. I was a musician for a while. I played in a rock band and toured all around the country before ending up going back to school, finishing majoring in biology, and then working at a medical school outside of Portland called OHSU. And from there, I traveled here. And so I've been here for about 3.5 to 4 years, and I work at the Harvard Museum of Natural History at the Museum of Comparative Zoology. Have any of you been there before? No. Me? Okay, so some of you have. It's a fun place to visit, so you can really go and check it out. It's pretty great. Lots of old animals and stuff. And so that's where I am. And now I'm going to just go over a couple things to review evolution with you, and then talk about development and talk about some research in these fields. Okay, so this is kind of a lot of stuff on this slide. But evolution by natural selection. This is the stuff that Darwin was talking about. Evolution works on mutations, right? On genes and how they mutate, and then how they're selected for in animals. And I think you guys have learned about this, but we've got a couple different phases here of evolution. And you start off with the starting material, which is your genes that you have in your body, or an animal or a plant has. And genes tell you different things. Genes can have mutations, or they can specify different things. And in combination with external factors like the environment, they help to set up phenotypes. So phenotypes, do you guys know what phenotypes are? Yeah? Physical traits, exactly. So phenotypes are physical traits, and they come about through being specified by the genes and by the environment. So if you have, what I mean by environment is if an animal has a gene to be really, really tall, but it doesn't get enough nutrition, maybe it wouldn't be tall. But if it was in the right environment, it would be tall. So it's a combination of these two factors. So in this example, we have a red and a yellow gene. And say they're specifying something, or they're setting up something, physical trait, a phenotype, like red or yellow fur. Now, red or yellow fur doesn't sound very fun, but suppose for a second that there's an environment that these animals live on where the red or yellow fur might help with camouflage. And if the environment suddenly turns from yellow to red, then those red ones that are really nicely camouflaged might not get eaten by their predators, and so they might be selected for. So they might not die, and they might survive to reproduce. And so that red gene might become more predominant in the population. And that's kind of the raw basics of evolution and how that works. Now, I know you've talked about some evidence for evolution, and so I'm going to go over that a little bit before we get on to talking about development. So evolution is a theory, and it's supported by an overwhelming amount of evidence. And I'm going to just talk about a couple really basic pieces of evidence here. And we can talk about more if you guys have questions later on at the end of the talk. But first I wanted to just ask why we might want to study evolution. Do you have any ideas about why we would want to even study this? Yeah. Yeah, go ahead.
See how humans evolved from monkeys.
Yes, that's a great reason. So to see how humans evolved from monkeys. Yeah.
To find the origin of life.
To find the origin of life. That's another great reason. Yeah. Sure. So to study natural patterns and how they might change in the future as well. Yeah, that's a really great reason. And another thing that I'm really interested in is just the diversity of life. So we see all these different things. We see fossils, crazy giant fossils. We see whales that swim underwater. We see plants, giant trees, and they all have evolved to be that way. And so by studying evolution, we can help to try to understand and explain this diversity of animals. And human origins is really important. People are very interested in where we've come from as a species. Okay, so a couple quick things to review evolution. These are things that should be a little bit familiar to you guys. First is, this is now a little bit washed out, so you can't see it very well. But the fossil record, so that's great evidence for evolution. So you see fossils throughout time that show nice transitions between one species or one type of animal and another. And what I have up here, which you probably can't see at all, is the transition from this little tiny dog-like animal to a modern horse. And that's a really nice incidence of evolution captured in the fossil record, because we have all the transitions between this little tiny dog and the great big horse that you know. And one of the really interesting parts about this story of the evolution of the horse is that it started off as an animal with 4 digits, and then it progressively lost those digits and started walking around on one of them. And in the fossil record, you can really nicely see that. You can see the loss of these digits. And so, since you can't really see this, I'm going to move on to the next point, which is homologous structure. Okay, so we have the horse limb here. And this horse limb is what we call homologous to other limbs and other vertebrate species. And what I mean by homology is that it has shared ancestry. And so this horse limb, which looks kind of weird, it's just got a single digit, right? It's walking around kind of like this. Really has the same structural basis as our hands that we have, or as the limbs of a dog. It has the same components, like it might have the radius and the ulna, or down in the leg it might have the femur. And it has the same patterns of the digits, the tarsals and metatarsals, and or metacarpals on the hand. And what is important to get from this is that these structures are all descended from a common structure, which is the initial limb of animals that first started to walk on land. And they've specialized to use their habitats well. So for us, we have a really nice dexterous hand. We can manipulate things and do all sorts of things with it. Horses are really out of luck when it comes to manipulating stuff, but they're really good at running on their limbs. Yeah. Yeah, if you guys have a question, you can go ahead and raise your hand. Yes? Oh, digit is a finger. Yeah, or a toe. Okay, another great piece of evolution or evidence for evolution is the geographic separation of species. And so this is kind of an important concept to understand, because it's about how species might form. And part of it is because they form during times of change over the Earth. And so you might have sorts of geographical barriers or geological barriers that arise that might separate a single species or population into 2 different ones, and then they might diverge. And in this population, we have some beetles here, that this initial starting circular population gets separated by this river. And then over years and years, you see that they differ a little bit. And so there's some green ones that arise here. Until finally there's a green and a brown population that are very different and they can't mix with each other anymore. Yes?
So are you saying that evolution happens over a long span of time, or one event happens on Earth that causes a sudden change?
Right. So yeah, there are two different things. So there's this geological event, say a mountain range comes up, which takes thousands or millions of years. And then there's evolution. And both of those usually happen over a long, long period of time. Yes?
So an event like, say, the Ice Age could be the reason why we exist?
Say again, the Ice Age, what?
Like you know the Ice Age?
Mm-hmm.
So an event like that would be the reason why we exist?
Yeah, so it definitely sets up the geographic pattern of animals. So animals had to come down south during the Ice Age because the glaciers moved south. And then they reproduced there and changed and differed. And so just testing out the microphone.
So since we're asking lots of questions, this is really good. So we'll just wait for the mic to get to you before you ask a question, okay? Thanks. Sorry.
Yeah. Yeah, so the Ice Age would be
Turn it off.
So we'll get that fixed. Okay, so yeah, so geography is really important. No problem. Okay. Well, maybe we won't use microphones. I can hear you guys okay, and I'll try to repeat your question. Yes. Yes. Yes, they did. They did, and I'll talk about that. So, in addition, there's a nice other piece of evidence for evolution, which you've also talked about in class, and that's similarities of embryos during development. And that's what you're referring to here. The pictures up here are two different embryos. I've got a lizard and a human embryo, and they look really similar, right? They don't look very different, even though lizards and humans look really different when they're adults, right? Okay, so the reason this is because during embryonic development, which we'll talk about in a second, which you've learned about a bit, all of these embryos pass through a certain stage where they need to really tightly regulate what's going on with their formation and growth. Oh, no problem. Okay. No, that's fine. No big deal. So what you start off with when you see embryos, they look really, really different. So this is a frog embryo here. This is a chick or a human embryo. I'm not sure. This is a fish embryo, and they all look really different. But then they start to develop, and they pass through the stage where they all look a little bit similar. And then after they develop and become adults, they look very different again. And so this similarity here is because of their shared ancestry. Because this is a really crucial period of their development where they need to set up lots of things about their body axis and make sure that everything is functioning. Okay, so the last little overview piece that I'm going to talk about, I'm sorry about the mic, hopefully you can hear me, is vestigial structures. And this is a nice and really intuitive way to look at evidence. So you can also see evidence for evolution in the formation of these structures called vestigial structures. And these are things that you kind of are familiar with a little bit, but I'll go over them. Snakes, as you know, don't have any limbs. They crawl around on the ground that you find, but they don't have any limbs. But in certain snakes, you can find that they have vestigial elements of their limbs left over. And so in this python, these two little bars down here are parts of its pelvis. And that pelvis was lost many millions of years ago, but you can still find these little bones that are present there that are evidence that it used to have it, that it descended from an animal that did have a pelvis. So doesn't really have a really major function. It stores some bacteria, might help break down food, but it's become relatively vestigial and not very important. Another example, which is similar to the snake example, is the whale pelvis. So whales also have lost limbs, but not their front limbs, just their hind limbs. And you can see little elements of that left behind with some whales. Okay, so this is all some evidence for evolution. And now I'm going to transition into talking about development and developmental biology. Right here is an embryo. This is a little movie that I'll show in a second. But first I wanted to ask, what is developmental biology? Does anyone know what developmental biology is and what we would study? Okay. Oh, yeah.
Well, it's like the growth of organisms over time.
That would be a great definition. I like that. And so it's looking at embryogenesis, which is the embryo turning from a little single cell into an adult, or the growth of organisms over time. So how they are formed from a single cell, how animals are formed. Development often involves a lot of complex movements and tissue rearrangements. These things are governed by genes. And so they're the same genes that evolution can act on to create new phenotypes. And we'll look at some of these movements and rearrangements of tissue later on. It's really important for how animals are formed. And why might we study this? Can anyone think of any reason why we want to study development? Yes?
To prevent mutations.
Yeah, that's probably the top reason. So birth defects are a major problem. And birth defects are defects of development. There's also a couple other things that are related to developmental biology and that are influenced by the study of it. So that's studying stem cells, because stem cells are really important during the development of organisms. And they're also really important for regenerative medicine and treating diseases. And so it's a really big field of research right now. Regeneration, so the salamander that I have up there that was on screen earlier, if you cut off one of its legs, it'll grow back. Why doesn't that happen in humans? No one really knows, actually. So it's not really normal. So that's the study of regeneration. That's also related to development. And also cancer. Cancer is a disease of genes and cells gone bad. And by looking at development and how cells and animals normally form, we can have a better idea of what causes cancer. Okay, so this is just little embryos moving around. It's a salamander embryo, and these are the ones that I collected in the field. I'll tell you a little bit about that too. Saying, yes. Well, this basically come out and say, hey, I'm here, I'm hatched. Okay, yeah, sorry, go ahead.
What was the circular?
The circular thing on the outside. So that's its chorion, or it's the membrane that protects it from the outside world. And so when they're laid in the wild, they need something to help protect them. They don't develop inside of a womb like a human would do. They develop outside. Yes. That's the yolk sac, because they don't get nutrition from their mother directly. They don't have an umbilical cord, and so they need to have all the yolk they would need to develop and grow into a little hatchling. It's like a chicken. Chickens have yolk and all the stuff inside the egg because the little embryos need that to develop. Okay, so I'm going to go over a couple different models of development, things that we use to study developmental biology. One is this nematode worm, which we call C. elegans because the Latin name for it is pretty much unpronounceable. Does anyone want to try? Yeah, I pretty much can't spell it out. You want to try? Or do you have a question? That's pretty close. I think that's the closest I've ever heard, so that's great. And it's a really, really simple animal. It develops from a egg, like all animals do. And when it's an adult, it's very, very, very simple. It only has 959 cells, so only under 1,000 cells in its whole body. And we know what all those cells do. And in comparison, a human might have over really nice development that you can look at visually and see. Closer to ourselves, the mouse, right? And so that's important to study because it's a mammal, so it's closely related to us. So if we're looking at diseases and things like that, it's good to look at a species that's close to us so we can help to translate that into treating our own diseases. Okay. And how do we even look at these embryos? So you guys have experience with some microscopes. They're small. Embryos are small, unless it's like a really giant, like a blue whale embryo. That might be a little big. But I even have some embryos here. We can look at later. And they're small. They're really tiny. So to be able to see the embryo, you can see it with your naked eye. But to see different features of the embryo, you have to look at our microscope. Salamanders.
Oh.
There's a black spot. The jelly stuff around it are the yolk, or not the yolk, but the membrane that helps protect it. Okay. Yes. Oh, I think it's fine. Yeah. Okay, yeah. Question? You don't have that. Okay. Yes.
Oh, what was that in the thing?
Okay, so those were salamander embryos.
Salamanders?
Yes, salamanders. That's what I actually studied, so I'll talk about them a little bit. They're amphibians. And the embryos of this dolphin we'll look at later on. Yeah.
Oh, sorry.
No problem. Okay, so to look at those, we use microscopes. And they can really range in price. So you can have a $100 microscope that you get off Amazon, or you can have a $500,000 microscope. It looks at fluorescent images, and it reconstructs things in 3 dimensions and all kinds of fancy stuff like that. So it really ranges. What else do we look at? We also look at genes. And so you guys have talked about genes. But we can visualize where these genes are inside of animals and inside of tissues. We can also look at the amount of them that's present over time, over development. This is just a movie of some genes. They're actually proteins in a piece of tissue, and we'll zoom in on them, and we'll just look at where they're found in the cells. And the blue is DNA, and the red is the tissue. It's kind of origin. So we're circling around, and we're looking at where it's found in this tissue. Okay, so just a quick overview of development. And then I'm going to show some movies of different aspects of development to help reinforce this. So we all start off as a single cell. Once the egg and sperm come together, you get fertilization. And then you have to start building up the basis of the body. So you have cell division, lots and lots of cell division to build up a lot of cells. And then after you have this nice starting material, you go through a couple of phases. One is called morphogenesis, and the other is called organogenesis. Those are kind of long, complicated words. But morphogenesis is taking the word morphology. Morphology is like the phenotype or how an animal looks. And genesis is it's kind of like development, so how the morphology is generated. And so during morphogenesis, you get a lot of tissue rearrangements. The animal will go through lots of complex movements when this expands, it blocks off other sperm from coming in and fertilizing it. And if you get multiple sperm fertilizing it, you might have some defects in the animal. This is the worm I was talking about earlier, C. elegans. And this is a picture or a movie of C. elegans right at fertilization. So you can see the DNA, which is going to be green because it's labeled fluorescently. Fluorescence comes from proteins that are found in jellyfish or sea corals. And when you shine certain wavelengths of light at them, they glow. They glow in the dark. And you'll see, here's one set of DNA in another. So this one might be from the father and this from the mother. You'll see them meet up, and then they'll undergo mitosis, they'll divide, and you'll see a couple of cell divisions. So here are the two pieces of DNA. Come and they meet each other, and then they're going to split apart here, and then this is the first cell division of the embryo. And you'll see it happen again. So here's the DNA divided again, and it'll happen again down here. So this is really the first part of life, first couple minutes of life. Okay, so then after, oh yeah, go ahead. By the shrink, it kind of compacts. It has to go through some different conformational changes when it is replicating itself. And so I think it goes from a very relaxed state to a more compact state before it pulls itself apart and divides. Okay, so then after we have fertilization, we need to go from a single cell or a couple cells into many, many cells to form an organism. And this again is the sphere, which is just a really nice, simple animal to look at. And you can see the first couple of cells in this here. So each of these is a cell. So it has 4 cells at this point. And what are these little dimples? What would those be? Yeah. Yes, you. Where the DNA is, right. Yeah, so it's the nucleus. Yeah, that's correct. So we can see these cells start to divide. Nice and synchronous. I don't remember if it goes through another cell. No, that's just going from 4 to 8 cells. So it keeps doubling like that. Okay, but it can't just keep doubling. It would just turn into a big mass of cells that literally have a shape to it, right? So that's where you need morphogenesis. And actually, I've got another picture of it. This is a different animal, and it's a bit of a different view. So this is an embryo of a zebrafish. It's a really common fish. You see them in aquariums or in pet trade, and it's really common to use in the lab because you get lots of eggs. This is the embryo. There are 2 different parts to this embryo. The top part is where most of the body will form from, and the bottom part is going to be lots of yolk, which will sustain the embryo through its development. And this video is kind of taking a slice through this spherical embryo, just looking at it as if you had cut it in half. And so what you'll see is lots of rearrangements in the yolk. But what's happening up here is cellular. So this humps, that's a boundary between 2 cells. And here's the second part of cell. Now you can see 4 cells. And now it's going to divide again. And it's really bouncy, right? It's doing all kinds of weird stuff. And the yolk is really getting rearranged. So the yolk is getting pushed into some of the cells to help the cells grow and nurture them. So any questions about that one? Yes? How long does the talk go for, or how long does that process take? The process, it took 104 minutes. So it took a little over an hour and 40 minutes. Yes?
Does it ever stop?
Does embryogenesis, does development stop? It does stop when you reach adulthood. Yeah. And it will change. It will look different over time. So lots of different processes happen. Okay, so now, yeah. Okay. No? Okay, can you guys hear me now? What about now?
Yes.
All right. Thank you. Okay, so now morphogenesis. This is a little bit more complex. It's got a lot of different movements to it, but it really sets up the whole animal and how it looks. And so, so in this example, this is a video that I took using really simple equipment. I just took a microscope and attached a camera to it and took pictures at a couple minute intervals over about 40 hours of development. And it's a video of this one type of salamander, the four-toed salamander. And so here you see the embryo, and this is this groove right here is where the nervous system will form. So I'll point out some different things. But these two little folds are coming together, and they're going to form what's going to become the brain. And you'll see the head start to develop over here. And so this part is going to become the head. This is an eye forming right here. So it's this kind of bulge that comes out. And then the mouth will be down here. And up here is where the limb will form. And so it's a lot of different things happening. This is the brain. This would be the back of the animal. And here you can see the eye a little bit better, right? And you can see part of the head. So it goes from kind of just a big ball of cells into something that looks like an animal. Yes?
Is the embryo like breathing or respiring?
Is it breathing? It's not really breathing at this point because it doesn't have things like lungs. It doesn't really need to breathe. It does do respiration. So there's probably some gas exchange happening. It's existing in water or in a wet environment. And so there's some oxygen coming in and carbon dioxide coming out because the cells themselves need to breathe. Okay, and then this is a second example of morphogenesis. And this is a zebrafish embryo again. And what the people have done here who've taken this movie is they've labeled half of the embryo in yellow and half of it in blue so that we can kind of see what's going on. This goes really quickly, but it helps you to see all the complex rearrangements that happen in this animal. So you see the yellow wrapping around. This is going to form the nervous system. This is the eye of the animal. This is the ear. So a lot of stuff happens. I'll just go through that once again. The nervous system up here. This is going to be the body. It's going to be along there. And that all happens within hours. It's really fast. Okay, then what about the organs? So those are important for you, right? You need your heart, you need your lungs, you need your stomach. This is not exactly the organs forming, but it's showing you the very early part in a chicken embryo. And what's going to happen is the researcher is going to inject some ink into the embryo, and you'll see the heart will pump it out to these blood vessels, which are on top of the yolk and helping to get nutrients from the yolk. Okay, so the little needle with the ink is going in, it goes to the heart, and then it flows out into these little blood vessels. And you can see the heart beating right here. It's pretty amazing. Yes? No, that didn't kill it. It's just a dye, so it can still survive. Yeah, so you can see it's nice heart beating. You can also look at embryos or pieces of organs in test tubes, right? And you can look at them develop as long as they behave themselves. And this is a mouse kidney. And the kidneys are really cool because they're these branch structures. They have a really complex morphology, or how they look, because they help to collect particles and toxins and stuff from the things you drink and help to produce urine. And so you'll see, as this develops, it branches, it becomes much more complex. So it's turning into kind of this tree-like arborized or branch structure. Yes?
What happens to all the fluids in the embryo after it grows?
After it grows?
Yeah.
You mean in reference to the yolk one?
Yeah.
The chicken one here? The ink will just, it'll go away, the embryo will metabolize it. The yolk, that will eventually be eaten up by the embryo through those blood vessels, and it'll use it to help grow. Okay, so we've gotten a basic overview of development and of evolution, and now I'm going to talk about combining both of these fields. So evolution is the process of going from really simple organisms to something very complex, and it's very similar to development, where you go from a single cell to a complex animal, like a squirrel here. Now remember, we've got evolution by natural selection. Genes and environment contribute to the phenotype, to the traits of the animal, which become selected on. What development is really interested in is the process through which you translate the genetic information into the phenotypic traits. And so it's just this little window. And you can use evolution to help study how development changes. You can also use development to help inform your ideas about evolution and how that is affected. Now, one way that we can apply this field, this Evo-Devo field, which is combining evolution and development, is to look at things like evolutionary transitions. And so an evolutionary transition is when you take one animal or animal form and it changes into another one through evolution. And I've got a couple of examples here. One is the example of a bird evolving from a dinosaur, right? And that's a very charismatic one. So what kinds of things do birds have that dinosaurs don't have? Yes. Birds have feathers. Who else? Yes?
Birds don't have any teeth.
They don't have teeth, right? They've lost their teeth. Yes. They're not mammals. They're different than mammals. Yes? They have hollow bones, right? Yes. Uh-huh.
They have beaks.
They have beaks. Yeah, beaks are really important. Back there?
Wings.
Wings. Wings. That's a great one. Okay, so I think we'll leave it at that. But there are lots of differences between dinosaurs and birds. And we can look at that in the fossil record, right? We can look at these transitional fossils that might be part dinosaur, part bird, right? They might have kind of something that resembles a wing. Or we can look at the development of these structures in modern birds and try to infer how that development has changed over time, or how we might actually be able to evolve something like a feather, or evolve something like a wing. And so, for instance, a wing is different than a dinosaur limb because you lose digits. So dinosaurs like a T-Rex is walking around like this. But they've got their 3 digits. Birds have kind of fused these digits together, and they have these long, hollow bones, as you said. And this is something you can look at over development, how this changes, and what sorts of genes might be responsible for changing a little dinosaur T-Rex limb into a long, majestic bird wing. Another great transition, which lots of people are interested in, is how we got animals that walk on land. So we are animals that walk on land, as are things like amphibians, lizards, crocodiles, and birds. But we evolved from animals that swam in the sea. We evolved from fish. And fish have fins, right? They have these floppy fins that don't do much. They help them swim through the water. But when these fish wanted to try to come onto land, they ran into a bit of a problem because they couldn't really walk on the fins. They weren't very supportive. They couldn't do things like manipulate food or hunt with them. And so the first tetrapods, or land animals, had to come up with limbs and these specialized organs for helping to walk around. And so by studying development, we've come to a better understanding of how you're able to actually make something as complex as a limb and how that changes through evolution. And so that's the fin into limb transition. Or you might go the other way around. Whales, as I mentioned earlier, they don't have any hind limbs. So how could we try to explain and understand how they lose these hind limbs? Whales developed from an animal that looked kind of like a dog, that was closely related to things like cows and deer. But when they went into water, they didn't really find themselves using their hind limbs anymore. Eventually, they went away. And so we can try to ask how that actually happened. What sorts of genes and mutations might be responsible? And so a couple little examples here. One might be familiar to you, and that's Darwin's finches. Darwin's finches are a group of birds which are found in the Galapagos Islands. And they're called Darwin's finches because Darwin had a big thing for them. He really liked them a lot. They're not very colorful or amazing to look at, but they have this diversity and variety of their beaks, which corresponds to the types of foods that they eat. And so you could imagine that a different type of beak or style of beak, the shape or the size, might help it to eat certain foods. Like, what would you do with a really, really long beak? What's something you could eat? Yes, something back there. Can you say that again? Wait.
If you have a really long beak, it could like get into small places.
Right, yeah, it could reach into small places. Yeah, yeah, go ahead.
An attack?
Attack.
Yeah.
Yeah, it can be defensive. Yeah, or if you have a really big beak, you might be able to eat really hard foods. And so that's what Darwin was kind of thinking about when he looked at these birds. And it was really helpful for him to come up with his theory of evolution. Because he said, well, there's all these birds here. They kind of all look the same, but they've got different beaks. I wonder what they're doing with these beaks. And what he came up with is that they're using these beaks differently to help interact with their environment and help feed or do things like self-defense. And so here are a couple different finches. We've got the large ground finch, which has this big, robust beak. And we've got the medium cactus finch, which has this longer and skinnier beak. And so a cactus finch, as you said, it uses its long beak to try to probe into flowers, and it will eat fruits and flowers with it. While these ground finches, they like to eat seeds. But the types of seeds they can eat vary depending on the size and the robusticity or the kind of strength of their beaks. And so if you have a medium ground finch with a medium-sized beak, it can eat small seeds. But if you have this really big beak here of a large ground finch, it can eat giant seeds, and it can really crack them well. And that's a great system to look at evolution, right? You can look at the evolution of these different beak sizes and shapes from a starting initial ancestral animal. But you can also look at development and how the developmental genes and the embryogenesis or developmental biology might help to set up how that beak looks. And so these beaks, as I mentioned, vary along a couple different axes. So you have the length axis, you have depth. The length is long, like Pinocchio shows long, and depth is kind of like the height, so how big and high it is. And so for the cactus ground finch, it's long. So this is the green bar here that says it's green and long relative to its depth, which is pretty short depth. In the medium one, it's not very long, so it's kind of a short beak, but it's medium height. And in the large one, you see a very large depth or height and a short length. And what researchers at Harvard have found is that each of these dimensions is actually controlled by a single gene. And so by changing the way these genes are present in the embryo, you can actually change the dimensions of the beaks. And what they show is from an initial starting type beak where you have low amounts of these genes, low amounts of the depth gene, low amounts of the length gene, you can vary these shapes just by changing those genes. So the length gene in this one is high, so you get a long beak. The length gene is low here, and the depth is high, so you get this really robust beak. And so we can look from the genetic level all the way up into the phenotype and see how the genes and how the development is influencing how these animals function in their environment, what types of food they can eat. Okay, so now I'm just going to talk in the last few slides about my own research, and then I'll take questions and I'll show you some animals. Okay, so I work in herpetology, and herpetology it's not the study of sexually transmitted diseases, it's the study of herps, which are amphibians, turtles, lizards, and then crocodiles. And so it's amphibians and reptiles is what people usually say. And this is a phylogenetic tree of vertebrates. It shows the common origins of these animals from a single starting ancestor, which would be down here, and then their diversification along an evolutionary lineage. So, for instance, fish are a very basic vertebrate. They swim around in the water. But then here, between fish and amphibians, we started walking on land. And so amphibians are kind of the first animals to walk on land. This is us here, mammals, like this now. And then you have this group of reptiles and birds. Okay, so herpetology is studying amphibians and reptiles. And it's important to study amphibians because they're this really basic group at the base of this lineage. It helps us to understand how animals descended from a fish and came onto land. It might also help us to understand other aspects of their biology, like regeneration. Like the one salamander that we have up here is able to regrow its limb. And so we can study that kind of stuff with it. My research is on a certain group of amphibians that don't have any lungs. And so from a starting ancestral animal, lungs were present. So when animals first came onto land, evolutionarily speaking, they had lungs. And so the common ancestor to all of us, all the animals that walk on land, had lungs. But in some amphibians, they've lost them. So they no longer have their lungs. Instead, they breathe through their skin, which we'll talk about in a second. Among these ones that don't have lungs, there's a group called the lungless salamanders, which is a very large and diverse group of salamanders. So when I say salamanders, actually when I say amphibians, what do you think of when I say amphibians? Frogs, okay. What else? Anything else? Yes? Yeah, go ahead.
Turtles.
Turtles. Those are not actually amphibians. Those are kind of closer to reptiles. Yes? Say again. Ponds, yes. They like to live in ponds. Yes.
Animals that can live on land at one point in their life and live in water at another.
Right, yeah, so that's kind of the definition of an amphibian. They live in both land and water. And so when I say amphibians, I mean frogs, and then also they've got other close relatives, which are salamanders, which are up here. These are all salamanders. And then there's also another really weird group of amphibians, which don't have any limbs. Yeah. No. Yeah, they're called caecilians with a C. And they burrow underground, and they're really hard to find. Not much is known about them. It's really actually an interesting area of study because no one knows anything about this whole group of animals. So salamanders are a little bit different than frogs. They're still kind of slimy, and they need water often to survive. But they have tails. They don't hop around. They kind of locomote as your pet dog or cat would. And that's what I studied. They're a very basic group of animals. And the ones that I studied don't have any lungs, right? So they descended from animals that had lungs, but they've lost them. They breathe instead through their skin. And this is how they do it. So this is a picture of a lung in a salamander that does have lungs. So lots of them do still have lungs. And this is a picture of the skin of a lungless salamander. And what these two tissues share is the presence of these blood vessels that go throughout them. And the blood vessels help to draw in oxygen from the outside environment and to exchange that with carbon dioxide. So that's respiration right there. And in the lung, in your lungs, for instance, you draw air in, and then there's little pockets of your lungs. And the capillaries and blood vessels in these little pockets help to exchange oxygen. So you get oxygen coming into your body, carbon dioxide coming out. It happens in a similar way in the salamanders. It's just over the skin. So they don't have anything to ventilate. They don't get to draw air into them. But they get to exchange oxygen over their entire skin because they have all these blood vessels and this thin and kind of fragile skin. And so for my research, I'm really interested in the evolution and development of this trait, this loss of the lungs. Because this is really weird. Why would you even want to lose your lungs? It doesn't seem like it would be very beneficial at all. And I'm interested in how they're able to do that developmentally and what genes might be responsible for that. And so to do that, I look at the embryos of these salamanders, and I have to go and collect them in the field because they don't breed very well in the lab. And so I look at a lot of different species. So this is the redback salamander, and I've got one up here, which I'll show you. Then a couple other salamanders, the dusty salamander and the two-line salamander. This is the four-toed salamander, and we collect these ones on Cape Cod, which sounds really nice, you know, beaches and warm and stuff like that. But really, it's actually not. This is where we collect them in Cape Cod, and it's basically the middle of a swamp, and it's full of poison sumac. So this is where I learned that I had a really bad poison sumac allergy, which is like poison ivy. And so sometimes you get to go to nice places, sometimes you don't. But we look for eggs. So here's my lab assistant looking for eggs. These are some eggs after finding them inside of a log. And what we do is, for each species of animal, we look in a different place to find the eggs because they have different habitat preferences, right? some of the females might lay their eggs inside a log, some might lay them in the water. And here's some that came from a log. And what I've shown so far and found in my research, this is a little Redback Salamander embryo, is that the lungless salamanders, they start to develop their lung, but then they lose it. So it's like they're giving a really good try. They're saying, oh, I really want to develop this lung, but then I can't quite get it to work and happen. So it starts to become there, but then it goes away. And so that's why I call it vestigial, because it's present, but in a very, in a not functional state, right? So then this vestigial lung disappears, it goes away, and that's it, then they become lungless. What I've also found is that the mouth in these salamanders might help to function like a lung. They might not only exchange gas over their skin, use the lining of their mouth to help to do gas exchange, to take in oxygen from the environment. And the way they do this is they have lots of blood vessels in their mouth to start with. And then they also express certain proteins that are found only in the lungs. They are found in the mouth of these salamanders, and so that's kind of weird. And they're able to draw air into their mouth through the nose and then ventilate like this. Okay, so kind of getting to the end here, but this is this vestigial lung structure in one of these lungless salamanders, and I'm going to show you a little video of it. So this is the salamander. This is a CT or a CAT scan, and this little tiny projection here is this developing lung. And here it is again. So the purple is just the kind of the mouth area and part of the gut, and then we're cutting away here, and this tiny little projection is what would become a lung in a regular lung salamander, but it's not present in the lungless salamander, and so it doesn't fully develop. Okay, so to summarize, Evo-Devo is the science of evolution development. It's combining these two fields, and it helps us to understand a couple different things. So I've told you about evolutionary transitions, like going from a dinosaur to a bird. I've told you about animal diversity, the beaks of Darwin's finches, and how those are able to change through evolution and change through development. And then also organ loss. So in my own research, I'm studying the loss of this organ, the lungs, which seems like it would be really important, but these animals are able to do away with it. So I hope that that's been a nice introduction to this field. I've got a couple different animals to show you, and then I'll take questions. I can take a couple right now, too, if you have one. Go ahead. Yeah, these two. So this is an axolotl, which I've got one right here. And they're from Mexico City. They're actually, they're almost extinct in the wild. But in the labs, they're really popular and common. And so they're an animal that's just a weird kind of twist of fate for this animal, that it's going extinct in the wild because of habitat destruction, but in the laboratory they flourish because they're a really common animal to use for studies. And these things on the sides of the head are gills. And so amphibians always go through a stage where they have gills. They might not use them, but they always keep them, or they always have them at a certain stage of their life. Yes?
How can the skin absorb oxygen?
So yeah, so the way that it does that is it's kind of just like a lung in humans. The lung in humans takes advantage of a couple things. It's very thin and delicate, and it has blood vessels that are very close to where the gas is coming in. And so in the lungless salamander, it is able to perform gas exchange, taking oxygen over the skin because the skin is very thin, and you have the capillaries or blood vessels which are really close to the skin. And so it's those two properties, just the proximity of the oxygen in the outside environment to the inside of the animal, the blood, that will help carry the oxygen to the rest of the animal. Okay, so now I'm going to show you a couple animals. First off, this guy, and this is actually, go over. And actually, could you get the lights too for this? Sure. Okay. So why am I turning off the lights? It's because this animal glows in the dark. Okay, and you can't really see it glowing right now. It's a little bit green. I'm going to try not to drop her, because that would suck. Sure. Okay, so I'll tell you about why it glows in the dark in a second. Now, can you see this? It might be a little scary. It's kind of a weird environment for it. Okay. So, and we can, if you want to come up after the talk, you can look at this a little bit closer too, because it's pretty cool. But what I've got here is a little flashlight that emits a certain wavelength of light. It's kind of close to like a black light or a UV light. And when it hits the cells in this animal, the cells fluoresce. And the reason they fluoresce is because this is what's called a transgenic animal. So you've probably heard about transgenic stuff, like transgenic crops and things like that. What it means is that the genes have been manipulated. And so the genes have been manipulated to put in a fluorescent protein which glows when you shine light on it. And that's present in all the cells in the animal. Questions about that? Okay. Yes?
Can you inject like human with that protein?
You could. Yeah, you could definitely do that. I think it would probably be pretty unethical. You have a hard time convincing someone to do that. Yes?
How do you know the difference between like the egg and the other egg?
The egg and the what?
Like, cause you said you went out in the field and you like found the eggs.
Oh yeah.
Yeah. How do you tell the difference between them?
Yeah. So they all kind of look very similar to each other.
Yeah.
So that's hard, except that the mothers will sit with the eggs and they'll guard them. And so then you can look at the adult and say, oh, this is an adult of this species. And so you kind of can know from that.
Have you ever got bitten?
No, they don't bite. These ones don't bite. They're very docile and very nice. This is one of the lungless salamanders, and so it's kind of small. Pull it out. And these are ones you can find just in the woods around here. They're redback salamanders. It's alive. And they're really cute. And they're not really that slimy. They feel kind of cool when they walk. And if anyone wants to hold it, you can afterwards. We have to be really careful with it. But they have a little red stripe on their back. They have a red stripe on their back, and they live in the woods. And so these are weird salamanders a little bit because they don't have an aquatic life stage. Amphibians, remember, means living in both land and the water. These ones, they lay their eggs on the land and they never go into the water. So they're perfectly happy just living on the land. Okay, the last set of animals I have here are frogs. You guys like to jump a little bit? And this is a Korean fire-bellied toad. You see it in red? Okay. So, trying to escape here. And the reason I'm wearing gloves is because these guys are a little bit toxic. And the red on her belly is meant to indicate that. So the red is a warning coloration saying to predators, don't eat me. I'm toxic and I don't taste very good. And this is called the Hanken reflex. It's turning on its back, right? because it's showing me its warning coloration, saying, don't eat me, I'm poisonous, right? So it's pretty cool. And it does that reflex when it's threatened by a predator. It will turn in a circle, turn backwards like that, like some sort of weird yoga position, and it'll say, I don't want to eat you. Okay. So, questions about that one? Yes? Would I die? I'm not sure. I'm not sure anyone's ever tried to find out. There are definitely some, so there are lots of amphibians that would kill you if you ate them, right? There are the poison dart frogs. There are some species of salamanders, some newts that are really, really toxic. And if you even ate a little tiny piece of them, you would die. But I don't think people would really worry about that too much because they don't really like to eat them. Yeah? You don't really get poisoned. It will secrete kind of a mucus, and then if I were to touch my eyes or to not wash my hands, then it might be kind of bad. It might just taste funny. Yes?
Did the poison dart frog and the pie-bellied toad, they ever evolve from a common ancestor?
They evolved from a common ancestor in frogs. They're not very closely related, but actually the fire-bellied toad is an important species to study because it's a very ancient line of frogs, and so it's one of the oldest and most ancient lineages of frogs. Yes.
What makes them toxic?
So they have these toxin glands. It depends on the animal. So with poison dart frogs, they actually eat poisonous ants, and they ingest that toxin, and then they are able to secrete it out. With these ones, I'm not actually sure what makes them poison. I think they produce the poison on their own. With some other animals, they produce toxins on their own. They're able to construct this poisonous protein and then secrete that out of the glands. And so toads, have all the warts on them, and those warts are mucus glands that help secrete toxins. Yes. What makes their color change? So these ones, they don't change their color in response to their environment. It's not like a chameleon, which might change their color. But they have a red belly, right? And that's pretty important and crucial for it. And so that's just something that develops that way. It's got certain pigment cells. Right, yeah, so they do like this. Yeah, so I mean that's one. So the question was, do other animals do that similar sort of reflex to help protect themselves? So the frogs, they go like this and they show their belly and they say, hey, I'm poisonous. Yes, there are actually salamanders, which are totally distantly related, that do the same thing. They curl up in a ball like that, or curl up kind of in an O shape and show their bellies. They might be aggressive. They might have displays, like they might pump up and down or something like that, to kind of show that they're potentially harmful. They have lots of different behaviors to show aggression or to show territoriality. Yes? I don't actually know. The question is, how do chameleons change the colors of their background? What they do have is lots of very complex pigment cells. So the pigment cells are able to kind of shift their coloration. But I don't know a good answer to that. Yes?
If the salamander has a little bit dark things with another salamander, will the offspring, will they glow in the dark?
Right, okay, so that's a good question. That's a question of genetics. And so you have two salamanders, say one is glowing and one's not glowing, and they're able to mate with each other, right? It depends on the kind of the structure of that gene inside of the glowing one. If you have just a single copy of that, then half of those animals will be glowing about, depending on some other different factors. But yes, it will produce offspring which glow. And so we use them in a lab like that. We cross the ones that are glowing with the ones that don't glow, and then we get 50-50 of glowing to not glowing embryos. Yeah. The black ones? Okay, so you can find them pretty much in any wooded area. I just lift up logs or lift up rocks and find them. and they're kind of all over the place. They don't come out a lot in the winter, but in months like in May and June and July, you can find them all over. And the ones that you commonly see on the forest floor are these little red newts, or orangish newts. And those ones are actually toxic, and they're showing you that they're toxic with their orange coloration. Yes?
Can the toxins be utilized for cures?
Right. Yeah, that's a whole field of medicine. So that's kind of, I'm trying to remember what that field is called, but actually my lab assistant is, he wants to become a doctor, and he actually wants to study that because he's always been interested in frogs, and he wants to take those frog toxins and help use them as medicine. Yeah, so it's really important to study them. And one of the actually most important things to remember about frogs and about salamanders and amphibians is that they are going extinct. They're going extinct very quickly, especially ones that are potentially beneficial, that might have really good good toxins that might help us treat diseases. And they're going extinct because of a lot of different factors, but climate change, human encroachment, so humans coming in and taking away their habitat, and also the presence of diseases that kind of set them over the edge. And so we really need to work to help save those amphibians in order to not only preserve the biodiversity, but also to help keep those potential cures that we haven't yet discovered. Yeah?
Me?
Yeah, go ahead.
I have two questions.
Yes.
Have you ever tried to mix two salamanders together?
Well, I've never tried to make two salamanders together because usually when you have different species, they don't actually produce viable offspring when you make them together. But we do things like manipulative experiments where we try to mix cells together. And so what I've done sometimes is to take, I'm interested in how the lungs are lost, and so I take cells from the lunged salamander and put them into the lungless salamander, and then try to see what happens to those embryos as they develop. And what was your second question?
What's your favorite type of amphibian?
Well, I think the ones I work on, they're really small, but they're really, really nice and cute. And so those are the lungless salamanders, like the little redback one that I showed you up here. They're just a really, really sweet, nice salamander, and you can just find them anywhere. So they're pretty fun. All right. Anything else? Any other questions? You're welcome to come talk to me at the end, and also to see the animals. Okay, one more question. So you're asking why would poisonous and non-poisonous produce successful? Well, it probably wouldn't usually. If there's two different species, usually there's what's called reproductive isolation. So they're kind of incompatible. They can't really mate together, or if they try to mate together, it wouldn't really result in anything. And so you can really only get things to successfully mate together if they're closely related. Yeah?
Why does the egg sort of look like a juvenile salamander with the
The things on the side. So so this picture here. So those are the gills. So these salamanders breathe both through their lungs and through their gills, actually, and also through their skin. So they're able to breathe through their skin. So they have three different sources of oxygen. And the gills are kind of like fish gills. They just help them underwater because they're just an extra amount of surface area to absorb oxygen from the surrounding water.
Are they fully developed or are they semi-developed?
These ones, that's a good question and a good point. These are semi-developed because usually in adults, they'll lose their gills. What's interesting about the axolotl, the salamander up here, and the one that we have over here, is that they never really fully develop. They reach this stage in their life where they're happy and swimming around in the water, they've got their gills, but they never take that next step and come on the land and lose the gills. And so they're arrested at this stage, and they're called neotenic or phenomorphic, and that just means that they didn't go through their entire developmental cycle. And then they are able to reproduce as a larval or kind of a young juvenile looking stage. Yeah? Do those what? Yeah, you can buy them at pet stores. They're pretty cute. Some people are horrified about them, but they're pretty cute. Yeah?
That red thing on the salamander, isn't that for it to sense where it's going?
It can sense where it's going?
Yeah, because aren't those blind?
So these ones are not blind.
They're not?
They have eyes, yeah. And where's my little pointer go? The eyes are here's an eye and here's an eye, and this is just the mouth. So they have a nose. They can smell, I think, a little bit, but they're able to use their eyes to navigate. And they also have other sensory organs on their skin, which helps them too.
Okay, so Mr. Lewis actually has a cab waiting for him. The next question will be the last one. So.
Yes. So I believe in evolution, and I think evolution is overwhelmingly supported by lots of evidence of fossils and the things I talked about. And that's kind of the field that I study in, and that's my background scientifically. And so I like to look at it from that perspective.